Welcome to the Everything Epigenetics podcast, where we discuss DNA regulation and the insights it can tell you about your health. Welcome to the Everything Epigenetics podcast, Alex. Super stoked to chat with you today.
Yeah, I'm happy to be here. Thanks for hosting.
Of course. I want to, I just want to know more about you. You're so interesting. You have a great background, and I'd love to have you explain to listeners, how'd you get to where you are today? Can you just tell me about your journey and what led you here?
Yeah, sure. So I was actually born in France, in the south of France. Moved around quite a bit when I was young, and then ultimately ended up in the U.S. In elementary school. Have been there pretty much since then. Went to college at University of Rochester in Western New York. Really cold place, but great. Really enjoyed my time there. That's kind of where I got into the aging space to begin with. I worked with Vera Gorbunova, who's a comparative biologist by training, who's really interested in the naked mole rat, which is the longest living rodent. And I also became very interested in this animal. It lives more than 30 years, which is incredible for an animal of that size. And started out doing mostly experimental work, a lot of cell culture. I was really interested in the hematopoietic system. So basically the entire immune system and how the hematopoietic stem cells become every other type of blood cell in the body. And then, yeah, I think I slowly transitioned more computationally. I studied abroad at University College London for a semester, took my first computational class there, and I realized there was just a huge amount of power to computational approaches in modern day biology. So came back, switched over. That's where I first got exposed to single cell RNA sequencing and a couple other single cell methods, which is I think what I work on now as well. And then yeah, after that, graduated in May 2020, and then moved to the Vadim Gladishev's group at Harvard, which was a lot of fun. I was there for a year and a half. Yeah, Vadim is honestly a really incredible mentor, and everyone in the lab is great. I think you just talked to Jesse Epigenetic recently as well from the lab. So it's yeah, really great place to study aging and really kind of the fundamental questions that are still unanswered. When does it begin? Why does aging happen? How does it happen? And really taking kind of a computational look at some of these things. And I worked specifically on epigenetic clocks, published this paper that we're talking about today, and as well as a couple others. And now, yeah, then I moved to Retro in January of 2022. I really wanted to. I think academia is great, but I really wanted to translate some of these findings into something that can be used in the clinic. I think it's a very powerful driving force for me to really improve the lives of people. And yeah, it's been an awesome journey building the computational team here, and we're really growing the company quite rapidly as well. So it's been a cool journey for sure.
That's great. And we'll talk about retro at the end for everyone
Yeah.
wondering, what's retro? I want to know more because I think you're right. Some people love the academia based side and they're very dedicated to that, but we don't get a lot of what am I trying to say? More of taking this to clinical use and kind of clinical practice. So super excited to hear about your experience there.
For sure.
And you're right. We're going to be talking about your great paper, the profiling epigenetic age in single cells, but we'll chat about
Yeah.
everything here and there. And starting with the big picture, before your paper was published in Nature in March of 2021, all existing epigenetic clocks relied on measurements derived from bulk samples. So meaning, and you can clarify this further, but basically samples that contain many cells, a saliva sample, a blood sample, a buccal swab. But you're taking a different approach. You're going to be looking at single cell epigenetics. So, what the heck is that? What caused you to do that? Why is this important? Give us all of the goodies.
Yeah, so as you mentioned, I think basically most of your listeners are familiar with this concept of epigenetic clocks of tracking aging, biological aging
Mhm.
through the methylome. Can also be done with any other modality, right? It's not exclusively the methylome. You could build transcriptomic clocks, you could build proteomic clocks, and these are some of the things that we're also working on at Retro. But yeah, to get to your question, I think there's something cool about getting like an age metric from a whole tissue. Like let's say you just give a vial of blood and you get your biological age for your whole body, or maybe just for your blood. But there's something also very interesting about how individual cells age. And I think that specifically the last decade has seen this this huge increase in interest, also in technologies to measure single cells. And you can do this across the epigenome, the transcriptome, and there are now multimodal assays as well that allow you to measure multiple modalities in the same cell, which I think is really incredible. And the beauty of single cell is really that you see the heterogeneity that occurs in any tissue. So this is particularly useful in tissues that have a lot of heterogeneity. So if you think about the blood, there are countless numbers of cell types, all doing a bunch of different things. And if you could understand how each of these individual cells age, I think it's just a really interesting kind of research question, and also has a lot of applications. So that's what kind of got me interested in it. I think there's obviously a lot of value still to these bulk measurements and bulk clocks. I think they're probably going to be more accurate for a while, but uh, especially in a therapeutic setting, when you're trying to figure out kind of perturbations or things that you can do to cells to rejuvenate them, you need to be able to measure the age of those individual cells. And so that's what really got me started into that SCH journey.
Yeah, and was that how did that idea start in the lab? Like was that you? Was that I know Vadim, Dr. Gladyshev,
Yeah.
just there are so many people I've connected with already after creating Everything Epigenetics. It's like you start making all of these connections, and it's just fascinating. So like, how do you even come up with this idea?
Yeah, this is definitely something that the Vadim was really interested in when I joined the lab.
Okay.
Um, and I also got very interested in it after talking to him before even joining. We had a couple of Zoom calls, and he kind of threw around this idea, and I was like, oh, that's very interesting. Like, why has no one done this before? Turns out that there were a lot of complications, right? So there was really minimal amounts of data to begin with, of public data to use, and it just required a totally new algorithm to work in the first place. But I think it was a really interesting project because it really bridges kind of this world of epigenetics and aging, which is just exploded over the last couple of years, with also some of these new technological advances that again enable you to understand heterogeneity and really measure things at the fully functional unit of life, which is a cell, right? So yeah, but I definitely need to give Vadim a lot of credit for it. I think he's a visionary, and he's been yeah, very impactful in my development as a scientist for sure.
Sure. Very exciting to hear about the work that has come out of his lab and continues to come out of his
Yeah,
lab as well. So when we're talking about the epigenetic age clocks, like you said, most people they're getting them from these bulk tissue samples. What are the differences then between the output of the bulk epigenetic sequencing methods and then the single cell sequencing methods that you did? And feel free to elaborate, spend a lot of time on this because,
yeah,
again, I think this is very foreign to listeners and really
yeah.
new. So I want them to be able to understand this.
Yeah, for sure. So most methylation profiling tools in the past have been again in bulk, and they just use these arrays. So it's a microarray. You hybridize some DNA onto that array, and whether it's methylated or not, you'll get a different reading, and so you can measure methylation based on that. But basically, all those individual reads that you're actually hybridizing to the array come from a bunch of different cells. And
Mhm.
so what happens when you're doing the downstream data processing is that you get a fractional value of methylation for every site. So every CPG, you'll get a value between 0 and 1, and that'll tell you basically how methylated that CPG is across a bunch of different cells in your sample. And the nice thing about that too is that you start with a lot of DNA. If you have thousands or millions of cells in your sample, you're going to have a lot of DNA. So you're going to be able to cover consistently a lot of CPG sites. And that's what most traditional clocks relied on. They relied on you take this array, you have values for each of these CPG sites, and you're able to train these kind of classical shallow machine learning models like ElasticNet that require kind of all these sites to be covered consistently. And that's great, I think. But in methylation, at the moment at least, it's really difficult to consistently cover all these sites. So you deal with a huge problem of sparsity, and this is common across all single cell modalities. It's improving all the time, but there's still a huge problem with sparsity. So not only do you get different sites covered in every different cell, but also the nature itself of the output is different. So again, in bulk it was fractional, so you can have like 30% methylation, 50%, 60%. But with single cells, it's theoretically more of a binary signal, right? Because that cell should just be on one strand. It should be either methylated or unmethylated. And you have exceptions. Obviously, you can have hemimethylation where you have one strand that is methylated and one that isn't. But in most cases, it's going to be a binary signal. So kind of dealing with the sparsity and the binary signal are the biggest differences. But there's improvements. I mean, This was in my paper. I mostly analyzed single cell whole genome bisulfite sequencing, so that's what produces the random reads across the genome. But if you do it in a more targeted fashion, and there's been a couple really cool preprints recently that kind of explore this idea, I think you could theoretically have a um, fairly similar matrix that what you would have in bulk. So it should enable even more methods to be able to be developed in that space.
Sure, sure. It's just amazing. Yeah, I like all of the graphics you use in your paper too. It makes it
Thank you.
really easy to understand. So for everyone who's thinking, what the heck is going on? I'll put those graphics and maybe like some explanations and stuff. And
Yeah,
of course, cite your paper. I hope everyone listening who's interested and wants to learn more actually gives Alex's paper a read because again, it's fabulous. This is just so cutting edge and really new stuff. Um, where where are you getting these samples from, like the single cells? Can you give me a little bit of background on that and how that works?
yeah, of course. So we were actually really lucky in that there was like not a huge amount, but enough public data to have a proof of
Okay.
concept kind of study, which is ultimately what we did. So yeah, we just used purely publicly available data. So there was one data set from Jan Vig's group that looked at hepatocytes, so liver cells. There was another great one in muscle stem cells from Wolf Reig's group. Wolf Reich, who's now Altos Labs, and he also has these really cool studies on embryogenesis, which is, I think, the crux of the paper, in my opinion, and also some of these embryonic stem cells as well. So, yeah, we were lucky to be able to mine kind of the public database, but I hope that more and more people, and this is something that I think a lot of people are interested in, is just generating more of these single cell profiles, higher quality sequencing, just to understand more and more
Mhm.
about these cells.
Yeah, and we're chatting about just that, the remarkable progress that there has been in these single cell omic kind of
Yeah.
profiling methodologies, I guess you can call it. So in your paper, you mentioned there's still some issues, some things that need to be figured out there. Can you describe some of those issues and then how you overcome some of the limiting factors or what you ran into? Because at the beginning you said, yeah, this is going to be really hard to do. You ran into some key points.
Yeah. Yeah, for sure. So I think this kind of ties back to one of your previous questions about like what's
Hmm.
different between bulk and single cell. And here again, it's mostly this binary nature of the data, so having to deal with zeros and
Hmm.
ones as opposed to a fractional value, and also just the sparsity, right? So in one cell, you might have one set of CPGs covered, and in another cell, you'll have a totally different set. And this
Hmm.
is problematic for kind of these conventional machine learning approaches that people had employed to build epigenetic clocks in the first place. So the way I tackled that was, on one hand, for the sparsity issue, is essentially like pre-computing and training models on bulk methylation to kind of map out how CPGs change in bulk across the age range. And this is all in mice, this
Hmm.
paper. Although I hope ultimately this is applied also in humans. That'd be really cool. And so you kind of like map out the trajectories of these CPGs. And
Hmm.
since you have kind of this pre-computed set, you can intersect that set with any new single cell that you get, and only look at the CPGs that are covered jointly within both of those sets. So you're not trying to kind of impute information of any kind. I explored some of these approaches, but they also weren't super mature at the time. Maybe in the future they will be. But so now you basically only use the data that's available and you map the single cell to this like reference data set of aging. I built these in a tissue specific manner. So I built one for the liver. Could also do multi-tissue and it worked as well, although a little bit less well because it's trying to learn um, kind of these tissue independent aging dynamics, which I think are harder than tissue dependent ones. And so that was for the sparsity, and then I think this also kind of solved the binary problem because basically if you have more methylation in an older sample, you expect to get more individual single cells that are methylated in that older sample
Hmm.
if you were to sample it again, right? And then
Yep.
conversely, if it goes down. And so you can kind of track how this goes and then estimate kind of like with a probability how close, how old it is basically compared to, yeah. There's a lot more details in the paper, but essentially
Yeah. Yeah.
you yeah, you just like, yeah. You map the binary methylation level with this probability that you compute, and for a single CPG that doesn't work super well. But when you aggregate over many age-related CPGs, you're able to build these really nice kind of Gaussian normal distributions, and then the top of that distribution, the maximum likelihood is ultimately the age that we kind of predict as the age of the cell. But obviously, there's a lot of like technical variations
Yeah,
and
yeah.
things like that. So it's not a perfect method by any means. I mean, if you look at some of the graphs, right, the box plots are pretty big. It definitely shows differences between young and old, and I think some of this embryogenesis stuff, like it was, is very significant. But obviously, a lot of improvements remain. And there was recently a paper, another preprint by Wolf Reich, where they kind of try to improve
Mhm.
a little bit on my algorithm, and then add some kind of feature selection steps as well. And so I think it's really cool to see people
That's
contributing
good.
and kind of improving on methods. Because I think
Yeah.
in one way, this is kind of the future. If you're able to understand how individual cells age and you're able to make really good metrics for that, you can totally start to evaluate a whole range of therapeutics that you otherwise
I
wouldn't
mean,
have been able to look at.
it opens up like another, like it's still epigenetics, but it opens up an
Yeah.
entire different field of epigenetics, like that hasn't even been touched. So I mean, again, that's why I was so excited to speak with you. So um no, I appreciate you really diving into the methods there. I know it gets a little bit more technical, so again, people can read through that if they want to learn a little bit more. And you said this was done on mice, right? Remind me.
Yeah, so this is the
Your
whole
study.
paper was done on mice, but theoretically you could do the same with
Okay.
humans. So if you're able to make
Okay.
these kind of reference data sets in humans, and then also get single cell data in humans, which there's much less single cell methylomics data than there is transcriptomics. I think single cell
Mhm.
RNA-seq is by far kind of the behemoth in the space, but now there's also a huge amount of interest in single cell attack-seq, a method developed by William Greenleaf and Jason Van Ruster at Stanford, who now has his own lab at Harvard as well. And I
Sure.
started of these like multimodal assays too, I think are like super exciting, right? If you can measure multiple kind of layers of biology in a single cell, it allows you to start looking at kind of like temporal dynamics and how like changes in the chromatin actually impact changes in transcription. And I think there's also a huge interest in proteomics, right? So the three layers, the DNA, the
Yeah.
RNA, and the proteins. If you can measure all three at the same time, I think you get a very comprehensive view of the cell that you were never able to get before. And I
Yeah.
think ultimately
Fascinating.
this will be really applicable to aging and any kind of rejuvenation research is understanding those multiple layers and building models and testing your therapeutics, your compounds, or your interventions on these and being able to evaluate them for the first time.
Yeah. Fascinating. Just yeah, extremely interested in again everything that you're doing.
Yeah,
So yes, let's keep diving right into it. This has
sure.
been great so far, Alex. So
Yep.
you basically developed this. Is it just SC age, single cell age, right?
Yep. SCA
Is
just the
the
name. Yeah.
SCH. So this novel clock method capable of profiling those epigenetic age in single cells. So I think you talked a little bit about how you did this and how you designed it. Is there anything else you want to add in terms of maybe the creation behind it?
Yeah, I mean, it was a really fortuitous kind of story. At the time in the lab, I was doing experimental work. I was working with a postdoc or instructor, Marco, who's now a professor in Spain, and we were really interested in selenoproteins, which is also a big interest of Vadim's. I think he's kind of pioneered that space in many ways, and to use some of the recoding mechanisms that are inherent to selenoprotein biology to try to address uh, uh, like stop code on mutations and then trying to read through basically those mutations. And these are involved in a lot of diseases in humans, and so it'd be really awesome if you're able to kind of cure them through this selenoprotein. Uh, yeah, like through harnessing selenoprotein biology and using kind of these other approaches to solve that. But at the same time, I was getting more computational and a little bit. And I think I got to the point where I had a nice proof of concept for this experimental project that I was doing. And then I was chatting with Vadim, and he was like, "Yeah, I think the single cell stuff would be really important to do." So then I kind of switched over fully to the single cell, and I toiled for a couple months with very minimal progress. I was trying all the conventional approaches, trying to get some of these imputation methods. I was trying a lot of things, and nothing was working. I think this really speaks to like how resilient one needs to be in science because for a long time things don't work. And then what happened is that I got an email in February of 2021 from one of my closest colleagues at the time, Csaba, who was a postdoc from Hungary at the time, is now a professor in Hungary, and he sent me a paper from a group in Germany by Wolfgang Wagner's group, And I read that paper, and the story behind that paper is also really interesting because it's a paper that Vadim had sent to another colleague of mine, Patrick Griffin, who works with David Sinclair. And then Patrick had sent that paper to Cheba, and then I had received the paper from Cheba. And this was late at night, I remember, in February. It was really cold in Boston. And I read the paper and I thought it was really interesting. And they kind of had some interesting kind of algorithmic ideas in there, trying to do some of these predictions based on like the lower number of sequence reads, which is ultimately what single cell is also kind of like compared to bulk. And I read it and I was inspired. And that very night I coded the very first implementation of SCH. So it improved over time, but the very first one was there that night. And I remember emailing Vadim the next day, and I was like, Vadim, I think I have something. And so yeah, and then like I worked really hard and got a preprint out in about a month, which was a marathon for sure. Then yeah, the review process lasted a couple months, and ultimately it was published in December of 2021 now. So yeah, it was
What a
very fortuitous.
story.
Yeah, very very thankful for everyone that was involved. I
Yeah,
think.
the perseverance. Like I'm learning a lot more about the computational biology and the bioinformatics and statistics and everything that goes in it. That's not my background. So I'm learning as time goes on, and definitely not even close to being an expert in it. Trying to train myself and learn more, but just the type of volumes it speaks about who you are, and people who take that up and dedicate their life work to it, because you're right. Like if you run into this problem and you're coding and I mean you can't figure stuff out, it's just like it makes sense to come together as a community and have all of these codes public and
Yeah,
kind of the moment when it clicked for you, that feeling has to be so good when it like actually happens and you sent that email. I'm sure that was
yeah,
like no other feeling.
yeah. It was definitely a beautiful thing because for a while I was getting not the greatest results, and so it was. But that
Right.
happens in science, right? Like it's usually work a while, and then sometimes there's a little bit of a breakthrough, and then things change, right? So
Yeah.
yeah, it was a
Yeah.
beautiful story. I'm super glad for I'm super happy that Vadim gave me this opportunity. I think it's amazing. Yeah, it was awesome.
Good. Good. Yeah. No. Thanks for sharing that.
Of course.
That's great to hear. Makes me smile. Well. All right. So, what's the application of the SC age? I think people are most
Yeah.
interested in like the application based questions because they just want to know like what does it do? How do I change? How do I get healthier? Or, what is it? How does this benefit me? Is what people are thinking.
Yeah. So I think there's a lot of applications, and it really
Hmm.
opens up like a whole new kind of area to look into. So I think some of the applications that we explored in our paper are really just the beginning, and I hope that people kind of look into a lot more things. But one of them is like so we first looked at these hepatocytes, which are the most predominant cell type in the liver. These produce albumin, and they're kind of crucial cell types. And so when we applied the tool to young and old hepatocytes, we saw that they aged, right? So the old ones were predicted to be younger than the young ones, which was quite cool because for some time people thought that there was a huge bias of cell composition in the clocks. And I think there still is to some extent, unless you explicitly correct for it. And there's some really great work recently by Alan Tomusiak at the Buck looking at some of these
Hmm.
differentiation independent methylation patterns and harnessing those to make age clocks that are independent of cell composition. But um, yeah, I think like it was for the first time like some evidence that individual cells themselves age because these hepatocytes, the older ones, had a different methylation profile than the younger ones, and judging by these age-related trends, there seemed to be something there. Another interesting application was this muscle stem cell data. So it looks like, at least epigenetically on an aging spectrum, the muscle stem cells don't age very much. And this had been already shown by Wolf in his original paper, just not at the single cell level. So we just kind of extended that to the single cell. We looked at embryonic stem cells too, and it looks like depending on how you culture them, there might be some differences too in their biological age. Although again, this is a very nebulous term, and I think people should not over-index on kind of these metrics. They are very important, and I think they will perhaps revolutionize kind of the longevity space. But there still needs to be a lot of work done in there for sure. But I think the most interesting one is kind of the last two figures of the paper, which we're looking at embryogenesis. And so just a couple of weeks before I was starting to work on my paper, my colleague Chaba, who I mentioned, he was doing some really cool work on applying conventional epigenetic clocks to embryogenesis data. So this is the period after fertilization all the way up to basically birth, but really the first couple of weeks after fertilization. And there was a theory that Vadim had kind of put out there in an opinion piece that perhaps cells rejuvenate during this period of embryogenesis, because if you think about it, when people have children, the gametes, so the sperm cells and the oocytes, they're old, right? They've aged with the person. Maybe they've aged less because they're a little bit less metabolically active, but they've still aged because they're in the body. They've been there. The stem cells are aging, et cetera, et cetera. So somehow they age, but they need to give rise to an embryo every time that is of age 0. Otherwise, every generation, every subsequent generation would get older, which doesn't make any sense. And we're not seeing that, right? Um, and so kind of mapping when that rejuvenation process happens during embryogenesis is a really interesting task. And Chaba did some really good work on that, doing it all at the bulk level. But for some of these like really early embryogenesis time points, there are just so few cells that it doesn't really make much sense to do it in bulk. And so one thing that we tried was using one of Wolf's data sets, and this is a fantastic Nature paper that he had published in 2019. We applied the method and we saw very similar trends. So basically from embryonic day 4.5, so 4.5 days after basically the formation of the zygote, we see kind of from 4.5 to 7.5, we see a really nice decrease in epigenetic age. And I think that was another kind of hint that indeed maybe this rejuvenation period does occur. In mice and probably in humans as well, because otherwise it'd be difficult to explain how we're able to reset age essentially. And I think it's like
Right.
super pertinent, specifically when people talk about like reprogramming and rejuvenation, because our body
Hmm.
naturally does this. Like we're trying to use some of these artificial methods. And I think the Yamanaka factors, the OSKM are a great
Hmm.
start, but they were they were artificially discovered, right, to turn fibroblasts into iPSCs.
Hmm.
Whereas here, our body like naturally kind of has this reset of the entire epigenome, and somehow that seems to be correlated with the decrease in epigenetic age using clocks that are only trained on normal aging, right? So not at
Hmm.
all in any embryonic data. So you just apply kind of out of distribution, and you still see these trends. And I think several people have kind of substantiated these results now. So I think it's just very interesting to kind of explore that. And I think single cell is a great way to look at those things. And but beyond that, lots of applications. I mean, you can think about maybe discerning if some cell types age at different rates than other cell types, if some cells in a distribution are older than other cells. If you give an intervention to some cells, maybe some of them get younger, maybe some of them don't. Understanding why that's the case. I mean, a lot of it opens up a whole Pandora's
Yeah.
box of just kind of a lot of
Yeah.
applications.
List could go on and on. Like I said, it's like a field within itself. I think the single cells and well, I mean, the immune cell deconvolution and all the work they're doing at the Buck Institute, yeah, it goes hand in hand with this a little bit, right? Really
Yeah,
identifying down to the cellular level, why are these markers different? How do we go through that reprogramming process? So just so many questions,
for
and
sure.
that you all are working on and chewing off and trying to get the answer to. So we'll see what comes from it all.
Indeed.
Our I know kind of when we were talking about bulk measurement,
Yep.
and then the single cell measurement, you talked about some of those limitations already. But would you say that there are any further limitations with your SC age that you would want to improve on?
Yeah, for sure. I think
Yeah.
you can always improve on these methods. I mean, one thing would be just getting better data, right? So
Yeah.
I trained all of my like reference models for aging based on bulk of like a whole tissue. So it was like bulk
Mm,
measurements of liver across different time points in mice. Ideally, if you only want to measure age in hepatocytes, it would be ideal to just have hepatocyte profiles. And maybe you're still doing these in
right.
bulk, but you've like sorted just hepatocytes, right? So instead of like, I mean, the liver is fairly homogeneous to most of these hepatocytes, but there are also Kupffer cells and endothelial cells. And so the composition of those might be changing. And I think some work from the Tabula Muris consortium indicates that there might be kind of some changes with age. So to abstract away fully from that cell composition and to stop it from biasing your results, I think it would be useful to be able to train those, but just
Yeah.
again, the data just doesn't exist. But I'm very hopeful that in the next couple of years, kind of people will start to make that kind of data of like cell type specific methylation profiles,
Hmm.
either in bulk that they can be used for single cell. Ideally, you would even want to train a single cell model to apply to single cells. But again, the sparsity
Hmm.
constraints at the moment are a problem. But there's new methods coming out, like RRBS, which is reduced representation by sulfide sequencing, which is a more targeted version. And you can even do even more targeted than that. So you can restrict it.
Hmm.
The TimeSeq paper from Patrick Griffin does a really great job of this, of using probes to restrict basically your measurements to only specific age-related sites. So if you're able to
Hmm.
consistently measure those, you could use more of these conventional approaches that people have tried in the past on single cell data, which would be great. And I think again there are also probably algorithmic improvements that can be made in feature
Hmm.
selection. And some of these have been tried by Wolf's and his colleagues in his most recent preprints. But yeah, there might be some additional things that we can do for sure. Can
Yeah.
always improve on these methods. I mean, if you look at single cell RNA-seq, really started about a decade ago, and just the absurd amount of methods that have come out now, and they each claim to be better than the previous. Not always the case, but there's
Yeah.
the tremendous amount of progress, both like on the data acquisition side, so just experimentally, but also on computational methods side. And I think the algorithm that I used was really simple. So there might be much more complex ways to get a better prediction for each individual cell.
Yeah, help reduce the noise of the algorithm and kind
Correct.
of
Yeah.
tie some things in there. No, that all makes sense. Well, yeah. What about the future of epigenetics in general? What's your your thoughts, your opinion there? Do you think? And I'm a firm believer, and everything has an application. The first generation clocks were created for a reason, right? When we talk about the bulk tissue ones, yes, they're trying to predict chronological age, but they were used to help date refugees to see if they were old enough to seek asylum, or for crime scene investigations from a DNA sample. So I think everything has an application and a use. But I guess what I'm trying to ask you is if you think the field of epigenetics in the future is going to be built more on the single cell clocks, or more of the bulk tissue methods. Again,
Yeah.
depending, I guess, on what you want to look at.
Yeah. Yeah, that's a tough question. I mean, I think again, there's value to both methods. I think
Mhm.
the more we the more we study heterogeneous systems, and the more we build kind of these perturbation screens or things like that that impact different cells in different ways. So like one cell might get one genetic perturbation, and another cell might get another. In those cases, you really
Mhm.
need single cell, because you're not really
Mhm.
going to understand anything from just averaging all the cells together. And I think this is a great time to bring up this classic analogy of the smoothie, right? So with bulk, you're looking at the smoothie of cells. If every cell is a fruit, you're
Mhm.
looking at smoothie, and that's what most clocks do. And you can tell kind of a young smoothie from an old smoothie. But if you're not understanding how the individual blackberries and blueberries and strawberries are, then you're missing a huge amount of information. And I think, yeah, like most of biology now is really moving pretty heavily into single cell approaches. Again, there are still huge constraints to them. They're still very expensive for many labs, although the cost is coming down every year, but it's still very expensive to
Mhm.
run. Quite a bit more technically difficult than bulk as well. And then the analysis too, a lot of batch effects, a lot of things you have to correct for. But
Yeah.
I'm hoping that kind of things move more and more towards single cell. And I think we're starting to see also some of the first clinical applications of single cell, specifically kind of in these high heterogeneity environments, understanding tumors and immuno-oncology and things like that. I think that's really where the promise of single cell lies. And again, you can do this across multiple modalities. And epigenetics, I think, is a huge one as well. So these bulk clocks will continue to be around. I think they'll be cheaper. They'll be like the TimeSeq
Mhm.
paper, for example, can bring down the cost to just a couple of dollars, which I think is really incredible from the
Yeah.
current like several $100s that you need to pay for one of these methylation arrays, and that's in bulk. But if you could have a version of it
Yeah.
in single cell, I think single cell is going to be much more of a research application than it is.
Mm.
Um,
Mhm.
yeah, I think maybe on a consumer level, it'll be much easier to do kind of these bulk epigenetic age measurements for a long time. But on a purely research base, I think understanding how individual cells respond to things and how they differ is also just like
Yeah.
really interesting.
Yeah, I agree with you on that. Love the smoothie example, by the way. That's great.
Yeah,
I'm going to make that a highlight so people can understand,
yeah,
when we're really digging into this. So no, that's a
yeah,
really great example. And I agree with you. Um, commercially, there's all these biological age clocks, and commercially, even like the biohacker consumer, or people who, are probably more well versed in the space than the general consumer, still may not understand the differences between all of these epigenetic clocks and what's going on, right?
Yeah,
So I think it's going to be harder to introduce the single cell to consumers, but I do think it is the future, especially as we're trying to understand causation rather than just correlation and what's
for
happening at
sure.
the cellular level. So yeah, I mean, it'll get cheaper as more people are using this technology and hopefully converting to that once there are more insights, those interventional trials and things. So I think it's
Yeah,
super fascinating.
totally. Yeah, in regard to
Um,
this causal argument that you make as well, there's a great preprint from one of my colleagues in Vadim's lab, Albert, who kind of starts to explore some of these causal relations, which are still totally
yeah.
kind of undetermined, right? Like, is
Mhm.
the damage in the methylome what causes aging in some way, or is it a consequence of aging, or this whole like damage hypothesis, like how does that link to these epigenetic clocks? Because we see very concerted changes in particular CPG sites with age. So is some of it stochastic? Is some of it not stochastic? And Andre, also from Vadim's lab, who's now at Retro, has explored some of these
Mhm.
questions. So I think there still remains a huge amount of mystery with how kind of aging and epigenetics mingle, but at the same time, it's really cool to see a lot of great work in that space.
Definitely. Yeah. Exciting nonetheless. But yeah, familiar with those papers and what you're talking about there. Um, so let's focus on you. What are you doing now? What is retro bioscience? Tell us more. I know there's been some things in the press, and I've been trying to keep myself updated, but would love to hear about everything you're doing.
Yeah, it's an amazing place for sure. Been really enjoying my time here. I joined really early, so the scientific team was just a handful of people, and I've been building up the computational team since then. We're
Hmm.
basically 8 people now, which is really cool. I think we just do do a lot and very complementary backgrounds as well, so I think we're going to be able to accomplish really awesome things. The whole company as a whole is about 35 people. We're based in Redwood City in California, and I think it just came out last week that Sam Altman is our investor as well. So he's the famed CEO of OpenAI and previously president of Y Combinator, the most legendary kind of incubator for startups. And it's really great to be able to do. I think one of the things that really defines Retro is well, there are many things. First of all, everyone's like super mission aligned and mission oriented. I think we're all just really interested in the problem of aging and how we can, at the root, kind of solve that, um, as opposed to just like going for like a particular age-related indication, right? Like how can we understand
Hmm.
aging itself and ideally make therapies that work against aging. We're also really hungry, so we really want to do this. Very kind of hacker, like scrappy culture as well. We
Yeah.
have a warehouse full of containers, which is kind of really unusual for a biotech. We're not kind of like the classic biotech like fancy lab with a bunch of windows. It's like
Hmm.
these like really funky like futuristic containers, but I think it's a great vibe. I think it's really awesome, and that was Joe's decision back in the day. Yeah, it's a really cool place.
The
We're
culture is
working.
what makes it. So.
Yeah, I think so too. I mean, it's ultimately what drew me here. I had interest
Mhm.
from many of these reprogramming startups and kind of longevity companies when I was planning to make my exit from academia. And when I met the people at Retro, I just knew this was the place that I wanted to be at. So yeah, we're interested scientifically in a bunch of things. I think reprogramming is definitely one of the big ones, and also to be able to do all the science in a fairly financially unrestricted sense is really great. Like I think in labs, I've
Mhm.
been fortunate to always have been in quite well-funded labs, but I know a lot of people who have a hard time kind of getting the experiments through because they lack funding or they need to write grants. So just having a huge amount of cash, in our case, $180 million to explore
Yeah.
this, is really clutch. And it just enables you to kind of be very creative. And if you have an experiment that you think would produce meaningful answers, you have the backing to do it, and even if it costs several $100,000. But more scientifically speaking, we're really interested again in reprogramming across a couple different cell types. We definitely have a quite prominent T cell program, so we're interested in rejuvenating T cells, which are a big part of the immune system, and they're involved also in not only vaccine responses and like regular responses to disease, but also immunotherapy, I think is a huge one. If you can improve immunotherapy by somehow rejuvenating T cells, that would be super cool.
Mhm.
One of the things we're interested in. We're also interested in other cell types, so we're exploring. We have programs in a couple other cell types as well. And then I think reprogramming, while it has the most potential, in my opinion, for really like resetting age and rejuvenating cells, should also not be the only thing that people look at. So we're also really interested
Mhm.
in how you can modulate the autophagy process in cells and how that can maybe be either rejuvenating or somehow delay or slow down aging in some way. And then also these plasma therapeutics, which is related to
Mhm.
some of the parabiosis work that was kind of pioneered by Irina Comboy and Tom Rando's lab, who now has their own lab in Berkeley, and also some work that I did, which actually just as of today is in press in Nature Aging. So looking forward to sharing that with people in the future. But
Yeah.
kind of how parabiosis, which is a really gruesome procedure when you link an old mouse and a young mouse and link the blood system for quite some time. People had done it for short amounts of time, but we were the first in Boston to do it for 3 months in collaboration with Jim White's lab at Duke. And then that showed some like rejuvenative phenotypes, especially on these clocks. It looks like a lot of kind of the hallmarks of aging were diminished as a result of being exposed to young blood, which is obviously not super practical for human applications. You can't
Mhm.
just connect a young person and an old person. I mean,
Mhm.
there's a great episode of Silicon Valley that is very much about that, right? Like these like blood boys.
Yeah.
But yeah, I mean, if you can figure out what specifically about young blood is good, like what maybe
Mhm.
youthful factors you can take or manufacture in some way to give to older people, or vice versa, what you can take from older people, like some of these negative, like old
Mhm.
factors in blood, and somehow remove them, all through kind of modulating the plasma proteome. I think that is like much more proximal to the clinic. And so that's also a really interesting kind of space to operate in, because it looks like this dilution hypothesis has some merit. There's
Mhm.
been a couple recent papers that shows that if you can dilute, if you can dilute plasma, you
Mhm.
may improve a variety of age-related facets. And so, kind of understanding what proteins are important there is kind of the crux of the equation. And so that's one of the things that we're interested in as well.
Yeah. Amazing. Yeah, I mean, I'm just wowed and yeah, just so excited about everything that you all are doing. Um, w- I don't know what I'm most excited about. Probably, yeah, I think what I spend most of my time on is, talking to healthcare providers about this epigenetic based testing in the integrative functional medicine space. And a lot of them
Yeah.
are doing like the young plasma exchanges or, uh, working with patients on yeah, figuring out what factors they can include for medication to try and make those clocks tick in hopefully backwards in the right direction, lowering your biological aging processes. So I think the application of that one is really exciting. I think it'll be as prevalent as like IV clinics are now. Like
Yeah.
you'll see them on every corner in like 10 years or something, and you could just like text your friend, hey, what's up, want to go get like young plasma exchanges, and it'll be a thing. So I think that's super exciting as well. Um, but yeah,
Yeah.
Alex, it was great to learn, about your single cell work, the clock that you created, even what you're doing at Retro. Is there anything else that you want our listeners to hear, or if people want to connect with you, where they can find you as well?
Yeah, for sure. Um, so first of all, Retro is hiring across multiple positions. So if people are interested, computational team is a little bit full right now, but we'll be hiring again probably this year. But all the other teams, all the
Mhm.
experimental teams are hiring. So if you're a scientist and you're interested in longevity and you really want to make an impact, yeah, definitely can't recommend Retro enough. I think it's one of the coolest places I've ever been in. Look forward to being here for quite some time as well. Uh, yeah, and I think um, it's been really awesome to see more and more interest in the longevity space recently. I think
Mhm.
like the formation of some of these fellowships, like the work that Nathan Chang has done at
Mhm.
ODLB, and now with Mark also at the Longevity Biotech Fellowship. So if people are interested in that, I'd recommend they check it out. It's just it's a way for people who come from non-traditional backgrounds, people who maybe haven't studied biology, to try to get into longevity. I think it's a really awesome effort as well. Yeah, more community, more awareness. It's been awesome to see also a lot of funding come into the space, but
Mhm.
also realizing that these things take time and they're really hard to do, right? So I think there's been a huge amount of hype with reprogramming, with the announcement of Altos and Ulimit and Retro,
Mhm.
but these things take time, and clinical trials and things like that will take time as well. but I'm optimistic that we'll be able to really have quite a powerful impact on
Yeah.
on longevity and on aging, and ultimately Retro's mission, right, is to increase healthy human lifespan by 10 years. And that's a super difficult mission, very ambitious, right? Because maybe like antibiotics and hygiene has drastically increased lifespan in the last like several hundred years, but like to
Yeah.
get to the point where someone is already healthy, like already exercising, doing all the right things for their longevity, and then giving them an additional 10 years of healthy lifespan,
Hmm.
right? Not lifespan um, where they're disabled or where they
Yeah.
really can't do much, where they're like bed stricken, right? I think like having 10 more years is super cool. And even if you can just get to 1 or 2 or 3, I think it'd be really
Yeah.
awesome, because like
I,
as
mhm. I think we'll start to see that soon is what I was going to say. I don't think it's unheard of. I think it's like super reasonable. I mean, it's not going to be easy, right?
yeah.
But like you said, with the team you all have and you're so mission driven, it's the motivation and the why
Yeah,
behind, all of your work that I think is just incredible.
for sure. And science is unpredictable too, right? So you never know really what
Yeah.
what will happen, but I think it's like fascinating research questions to work in, and hopefully we're able to translate them into the clinic within a reasonable amount of time. And I think that would be the most fulfilling, right? If you can see a therapy that
Yeah.
you were involved in creating, and it provides someone with like more time to spend with their family or more time to do some of their favorite hobbies, or I think it's
Yeah.
really cool. And I think also one last thing is, I think like the advances in artificial intelligence are also really really interesting,
Oh,
and the kind of mingle between that and longevity is potentially very powerful. I think I feel like
yeah.
some of the recent announcements from OpenAI, for example, GPT-4 this week and ChatGPT a
Hmm.
couple months ago. I think if you're able to kind of harness some of these like large language models, and also just like algorithmic improvements that have occurred in the last couple of years in the field of deep learning and artificial intelligence, you could really make quite fundamental dents into how you understand biology in the first place. I think right now our models and all our representations are pretty good, but they're not perfect by any means. And I think it'll
Hmm.
be hard to get anything perfect. I think biology is like very noisy, and finding the signal in the first place is really difficult. But it's a really exciting
Yeah.
time to where you can maybe leverage some of these advances in AI and merge them with biology, and also both from the experimental side, improve methods, and also on the computational side to just understand things better, and then ultimately uh, enable more of these therapies to come to light. I think it would be super cool.
Yeah. I think so too. I think that was well put. That's just like a little bow wrapped on our entire conversation. So one last question for you, Alex.
Yeah.
I ask everyone this at the end of the podcast. It's just a really fun question. If you could be any animal in the world, what would you be and why?
Oh wow, I wasn't ready for this one. That's a really interesting
Make you
one.
think.
I used to ride horses when I was younger, so I definitely have a good
Okay.
vibe with the horse.
Okay.
I also really like cats and dogs. I had a dog for 13 years, golden retriever.
Yeah. Oh, love
Uh,
Goldens. I grew up with
y-
Goldens.
yeah, they're great dogs. Let's see. Hmm. I like owls too. I think owls have a good vibe.
Oh,
Owls are cool. They
that's unique. Yeah, no one has said an owl yet.
they're just kind
Okay.
of like chilling in the trees, and they're kind of nocturnal as well. So I recently saw a llama
Yeah.
too in California. It was an unexpected sight
Yeah.
to see a llama, but I think I like to think about things. So maybe an owl would be a good one.
Perfect. But yeah, we'll put it on the table, but not confirmed yet. So
Yeah, no,
maybe an
no
owl.
confirmation, indeed. Yeah, and if people
Cool.
also,
Well,
sorry, just if people want to reach out to me
yeah.
as well, I'm on Twitter. Alexander
Hmm.
Trapp is my handle. You can find me there. And you can also just email me, alex@retro.bio. Feel free to reach out if you have any interests or questions, or especially if you're interested in what we're doing here. I think it'd be cool to chat.
Yeah, just to open up the conversation. Well, I'll put everything in the show notes. If anyone
Awesome.
has any questions for Alex, send them to me. If you can't find him, we'll make sure that they get to him. But yeah, thank you so much, everyone, for joining us at Everything Epigenetics podcast. Remember, you have control over your epigenetics, so tune in next time to learn more. Thanks so much, Alex.
Yeah, thank you. It was a pleasure.